Welcome to Longevity and Aging series. Today I would like to introduce Dr. Julia Sidorova. Julia is a research associate professor in the Department of Pathology at the University of Washington in Seattle, where she studies DNA replication and replicating stress, particularly in the context of normal versus cancer cells. Her research interests include DNA replication, genetic instability, and the interplay between replicative stress and the maintenance of genetic markers. Dr. Sidorova recently published a paper titled Werner syndrome, RECQ helicase participates in and direct maintenance of the protein complexes of constitutive heterochromatin in proliferating human cells. So my understanding of the paper is that it was shown that Werner disruption of Werner helicase basically introduces some disruption in heterochromatin structure. It disrupts protein complexes like HDAC2 and HP1α, and also downregulates LBR and Lamin B, which leads to derepression of some heterochromatin. So at least for me, it took some time to recall all the details of replication and heterochromatin. So I think it's good to define first, like, what is heterochromatin, why it is important in aging, and how is it related to replicative stress. Let's start with this.
Yeah, so thanks for this kind of additional intro. So what is heterochromatin? So generally, very kind of simplified speaking, it's a subtype of chromatin, which is kind of its most prominent feature is transcriptional silence. So, right, this is kind of the original defining feature. It's also structurally different, and that structure supports the silence. So there are two types of heterochromatin. One is kind of really deep silence, almost always, and that is called constitutive heterochromatin. And then there's facultative, which is sometimes silence. Some genes in some cells are transcribed, others aren't. So in development, throughout the cellular lifespan, facultative chromatin can open, close genes, and so forth. So now with constitutive heterochromatin, what it's dedicating to deep silence or committing to deep silence is mostly repetitive DNA. And so as we know, repetitive DNA takes up almost over 50% of the human genome. So imagine it's very important to control all that repetitive DNA and kind of keep it down. And so, so the function, why is it important to actually maintain heterochromatin is because essentially the deep silence that it provides not only sort of controls the transcription of the repetitive elements that it covers, but apparently it also affects how gene regulation overall in other types of chromatin. All genes are regulated. So that is really the kind of key functions of heterochromatin that is important. And so essentially, so what's I guess how now it's connected to aging. Now, the theory that we're talking about here is that of kind of relaxation, loss of heterochromatin during the cellular lifespan. As the cell gets older, apparently sort of kind of loses integrity of heterochromatin. That's really the model that is now supported by quite a lot of findings. And so, of course, one would say, well, why would sort of loss of heterochromatin kind of be associated with aging and even causative of aging? Well, imagine all these repetitive elements are now beginning to transcribe to be expressed as RNA at a sort of higher level. So apparently, what that actually achieves is indeed global genomic deregulation, loss of actually 3D architecture of the whole genome, not just kind of the heterochromatin parts. And I guess one of the most exciting probably of the last maybe decade findings is that such upheaval of these repetitive elements can actually be associated with increase in inflammatory signaling, both inside the cells that actually experience this upheaval and also outside in tissues. So that's really kind of the backbone of all of this. So and kind of let me see now. Should we switch to now WRN role in all of this? Kind of does that?
I think it would be good to also discuss what lamina is, because our pair regulates lamina, right, and lamin B interaction, and you showed that deficiency in Werner helicase basically disrupt specifically lamina related part of chromatin, right? So
yeah, thank you for that reminder. So what is the lamina and how does it connect to heterochromatin? Kind of why are we talking about it? So generally, lamina is kind of this underlining of the nucleus that is right under the nuclear membrane, right? And it is composed predominantly with the two types of lamins. One is called lamin A, really, a class, and then lamin B. So lamin B1 that we're talking about is one of this class. So this lamina is essentially kind of scaffold, which is really dynamic. It actually kind of can change its rigidity depending on the cellular needs. And what it does, it actually anchors the whole genome to the nuclear envelope as it is. And in doing so, it essentially creates or enables this 3D structure where kind of every region of the genome is in its place, in its proper specific place. And so now connecting kind of that to heterochromatin, well, heterochromatin is close to the lamina, and it provides actually the anchorage to the lamina, nuclear envelope as a result. So it's kind of the places of the chromatin where it's connected. And as such, it essentially enables, just as lamina, it's kind of part 2 of this 3D organizer for the whole genome. So that's where the kind of the interest in lamins and heterochromatin, that's what in aging, that's really the kernel of it.
And as I know, there are several progress related to lamina and chromatin structure. So maybe we can discuss also what is Werner syndrome and how deficiency in Werner helicase drives syndrome manifestations.
Yeah, so Werner syndrome. Werner syndrome is one of the progerias, and it's a rare disease. It's and by progerias, of course, we mean diseases or syndromes, congenital syndromes that accelerate aging. So people with these diseases essentially age, have not a normal lifespan, but greatly accelerate. And of course, everybody knows the Hutchinson-Gilford progeria, probably the most extreme one, where, of course, these essentially kids, 10, 17-year-old kids look like 80-year-olds. But a Werner syndrome is a much milder version of this, where people essentially achieve puberty normally, and then after that it becomes the speed of decline actually increases, and we're talking maybe people in their 50s look like 70, 80-year-olds. So it's a milder progeria, and of course has been described for a while, and then as with all progerias, great interest was in identifying the gene that is responsible for it. And so it was identified in the 1990s, and it turns out to be a helicase, a DNA helicase. So an enzyme that actually deals with unwinding DNA double strand. And so that was really a great surprise. Moreover, it's kind of what we call a housekeeping gene. In a way, it's everywhere. And it does a lot of things. It basically is involved in anywhere that DNA has to be unwound. It is involved in replication, it's involved in DNA repair, in recombination, telomere maintenance, transcription. And so really it provided a great kind of puzzle as to why this helicase. It actually also has, in fact, second enzymatic activity, which is called exonuclease. So it's a helicase that also can digest DNA. So why this enzyme? has such a strong sort of accelerating agent component when it's mutated, which is what happens when Werner syndrome results. It's basically a loss of this one single gene. So, and usually it's complete absence of it. So mutations in WRN, which is the gene, right, the Werner helicase, WRN is the kind of abbreviation of the gene. When it's mutated in Werner syndrome, there are lots of mutations, but they all end up in no protein. So the protein is completely lost, and that results in congenital syndrome. That's kind of the background on this.
Mm-hmm. And you mentioned that there are many functions like of Werner helicase and replication and repair. So what do we know about the mechanisms like of Werner syndrome?
Oh yeah, and so they kind of picking up that same thread that DNA helicase is involved in DNA metabolic processes such as replication and so forth. So that really kind of these studies that really pursued the sort of the enzymatic activities of WRN on DNA were some of the studies that kind of created this concept that, well, if you don't have WRN, you basically don't kind of work with your DNA properly. So you make mistakes, replication is not optimal, DNA repair is not optimal. And the result of this is what's called genomic instability. So in other words, the genome is just not maintained as well as it should be. So there are mutations, there are genomic rearrangements, there are loss of sort of genomic chunks. Kind of that's really the concept behind this genomic instability. And so the original hypothesis of how WRN can result in accelerated aging is that genomic instability can accelerate aging of the cell. Just things are just not, working well. Unstable genome cannot instruct the rest of the cell to work properly. And that, in a way, also kind of now oversimplified, once again, understanding of what might be happening. And in fact, the paper that we've published is kind of one of these findings that basically tell you that it's not all about genomic instability, but genomic instability, yes, it is connected to sort of aging, but that's not all, because there's also there are also functions of WRN that are kind of directly connected to maintenance of heterochromatin. However, maintenance of heterochromatin in and of itself can be connected to genomic instability. So it's really a complex kind of regulatory systems of these multiple circuits that sort of talk to each other.
Mm-hmm. And I think the other thing which is important for the syndrome manifestation is decreased replication, right? So which might be resulting in delay of the development.
Yeah.
So I think like one of the manifestation, I guess, also could be DNA damage, which leads to senescence. And what similar features we see and what is different in senescent cells in terms of heterochromatin changes and in Werner helicase deficient cells?
Yeah, so so essentially there are different kinds of senescence, and by senescence, so I use terms aging cells and senescence, and so so these are slightly different terms. A cell in its lifespan can exhibit features of sort of aging, that's kind of slow accrual of certain symptoms that are called actually hallmarks of aging. But senescence is really where all that accrual kind of results in this almost sort of transition into this different stable state, which is that state of senescence. And so many factors can actually push cells over the edge. And one of them is excessive DNA damage. So again, a kind of disruption of the genome that is above a certain tolerable sort of a threshold. The other kinds is actually oncogene induction. Certain oncogenes, when they are suddenly all produced in the cells, kind of push them into the senescence. And also just history of cell division, kind of the slow accrual. So that's called replicative senescence. So the question is, and the actual senescent state that is achieved after all these stimuli is somewhat different. For example, there are common features and there are different features. The most common feature is replication arrest, meaning the cell does not divide. The other feature is the cell is kind of in this hyperinflammatory state. It actually pumps out pro-inflammatory features, pro-inflammatory sort of molecules, cytokines. Now, what can vary is basically, I think, extent of the sort of this the composition of that secretory phenotype, the composition of these molecules. Also, the kind of ultimately the way the genome reorganized. Sometimes there are kind of very specific, very recognizable, what they're called senescence-associated heterochromatin foci. So it's basically just kind of collapsed chunks of genome that basically went from the surface into the kind of core of the from the periphery of the nucleus to the sort of almost like centrally located or no longer peripheral. Other times it's not the case. So the question is how absence of WRN resembles this state or doesn't. And I guess the answer is it's sort of when, and again, kind of this is what we've been seeing in the paper by looking kind of these different aspects of heterochromatin state in WRN deficient cells that are proliferating but are WRN deficient versus cells that are aging or senesced, fully senesced. And so my impression is that there's that kind of in a WRN deficient cells that is proliferating, the effects are milder. They're still there, but they're just sort of not as prominent. Some features are present as in a senescent cell or aging cell. Other features do not seem to be sort of as prominent. So it's really sort of at least as far as kind of we can see at this level, a quantitative difference, which is probably why WRN progeria, WRN mutation progeria, Werner syndrome is not such a prominent progeria. Basically, the lifespan again is affected, but relatively mildly compared to other progerias. So that's sort of I think that's kind of my take-home message from what we've investigated.
Mm-hmm. And but do you see this senescence-associated heterochromatin foci in the Werner deficient cells? I guess you.
Yeah, that's actually a very good question. So and the short answer is we do not, but that is because this particular form of three-dimensional rearrangement of the genome is actually not happening with kind of natural replicative senescence, at least in, for example, fibroblasts, kind of the cells that basically are, for example, that basically provide kind of a stroma for many of our tissues and are basically division competent. So they can actually divide they're not sort of differentiated cells, if you will, at least in that sense. So they're division competent cells that are underlie our skin, right? The stroma of many organs. So in these cells, these heterochromatin foci do not form as they replicatively senesce. And so where they do form is in oncogene-induced senescence, this very prominent response to certain oncogenes. And interestingly enough, we don't know much about how Werner absence modifies oncogene-induced senescence. That's actually kind of a surprising gap a partial gap in knowledge. So we looked a little bit into it, and it seems like if you try to induce oncogene-induced senescence in a WRN-deficient cells, it almost sort of, I guess I would call somewhat epistatic to WRN deficiency, because it's such a strong radical response that basically induction of oncogene-induced senescence is not synergizing, but rather is epistatic to WRN deficiency. So in other words, the heterochromatin foci form almost as if regardless of WRN presence. So kind of for one, that's one example of this epistatic kind of relationship. And for example, another example would be inflammatory signaling is very strong in oncogene-induced senescence. And it, again, is epistatic to the induction of inflammatory signaling that is seen in WRN deficiency, or virtually epistatic. It's really kind of very close.
Mm-hmm. And I noticed that in your paper, you show that H3K9 methyl-free marks increase in oncogene-induced senescence, but also in Werner deficient cells, but not in replicative senescence. Why do you think it's happening, and what could be consequences of that?
Yeah, so that's actually, I don't know. The short answer is I don't quite know. What I sort of can say is that just that generally the level of this particular methylated, trimethylated histone mark, which is actually mark that marks constitutive heterochromatin, but actually it's present elsewhere. It can be found also in facultative chromatin and so forth. So the relationship between this mark and, if you will, and aging is not sort of is not completely straightforward. Indeed, there are cases where it is lost. Other cases where at that kind of level, total level of this mark, it's not really affected. And that's basically example of replicative senescence versus oncogene-induced senescence. So there are studies where this mark is actually increased. If I'm not mistaken, in progeria, in Hutchinson-Gilford progeria, trimethylated canine trimethylated histone is actually up. So essentially, I think the answer is ultimately in kind of looking at more detailed level at specific areas of the genome, and basically seeing kind of that there will be some cases where, for example, if you do genome-wide mapping of this mark, you'll see increases and decreases in aging in senescent cells. And so there is this level of kind of detail there that we're not quite capturing yet in terms of why in Werner deficiency we only see, for example, mild losses on repetitive DNA of this mark. But then again, these are the losses that matter. So we see these losses that occur specifically on, in this particular case, particular subset of repetitive elements, and these are the ones that comprise the subcentromere, so it's pericentromere area. So next to the centromere. So these repetitive elements lose the mark in WRN deficient cells, whereas kind of generally a whole cell, we cannot detect the loss of this mark. So that's basically it seems like just these areas are kind of affected the most in this case.
Yeah, and talking about this particular area, SATII repeats, right, pericentromeric, where you see loss in heterochromatin organization in Werner deficient cells. Have you seen anything similar in senescent cells? Do senescent cells, replicative induced or replicative senescence or oncogene-induced senescence, do you see in the cells changes in SATII pericentromeric?
Yes, Absolutely, yes. So that feature is definitely shared in many types of senescence, particularly these the class of these repetitive DNAs called SAT2, satellite 2, in the pericentromere. That is a common feature of senescent cells, different types of senescent cells. So in this case, WRN indeed is definitely whatever happens in WRN-deficient cells also happens in senescent cells, only perhaps to a greater degree in terms of greater derepression, greater loss of heterochromatin on SAT2s, and greater derepression of SAT2s as a result.
Great. And the consequences of that in general are what?
Yeah, so the once again, kind of, and I should say that in many of these sort of answers, I'm not just, of course, I'm not talking just about our work. I'm talking about the whole field and work of my local colleagues at the University of Washington, as well as again, kind of many labs. But it is indeed a very interesting development in probably last 10, 15 years, is that if you derepress classes of repetitive DNA, including satellites, SAT2s or SAT3s pericentromere, you would think, well, what could go wrong? You just express some RNA. But the consequences are really global, systemic to the cell. Well, the simplest one, easier to understand, is, well, your centromere is not going to work perfectly. So you're actually going to have mitotic errors. For example, you're not going to be separating your DNA properly. Well, that can result in DNA breaking as cells divide. And now you have these chunks of DNA, chunks of chromatin sort of in the cell. And that actually has been shown to induce, under some circumstances, inflammatory signaling, because the cell responds to these chunks of DNA in the cytoplasm. So that's your first consequence. But there's actually kind of more, and in a way kind of less dramatic as this. And that is because apparently, particularly SAT2, actually, RNA, what it can do, it can regulate genome topography overall by titrating away major chromatin regulators that basically organize the proper spatial organization of the genome. So imagine now this RNA floods the cell, floods the nucleus, titrates away the factors. The whole genome can actually be dislocalized somewhat. I mean, I'm sort of exaggerating, but that's really the idea. And then as a result, you change genome regulation throughout. And it's been shown that things like SAT2 in particular can actually directly, if you overexpress it, can directly activate pro-inflammatory genes. So that's, again, kind of and that is, again, conducive and associated with aging. So in terms of genomic instability, SAT2, derepression of things like SAT2 can actually also increase genomic instability, because imagine you have essentially tandons of kind of, dozens of these copies. The longest SAT2 array and is about it's sort of almost megabase lengths. There, is it? I forgot, but basically imagine these are like tens of kilobases long, hundreds of kilobases long, in some extreme cases, of repeated DNA. If you begin to transcribe all these, and that's sort of the units, if you begin to transcribe all these units, you can sort of cause chaos in a certain sense, because they are, or let's say you begin to replicate it, and these are repeats that are also inverted, direct orientation and inverted. You can actually start having replication problems there, as well as kind of related to that, recombination problems, because now you have sort of repeated copies that can actually sort of slip, the DNA strands can slip and reanneal, and you got these sort of looped out repeats. So essentially, kind of the opening and metabolism of repetitive DNA is just oftentimes just a sort of a problem for replication and DNA repair and DNA recombination to deal with. So that's really kind of where this genomic instability comes in. And of course, you can again titrate away all the factors that normally deal with DNA replication or repair, because you're now having all this sort of these whole array that you need to deal with. So that's just kind of the idea there in sort of somewhat popularized terms, if you will.
Mm-hmm. Great. And you also show that this deficiency in Werner helicase, as we discussed before, leads to decrease in LBR and depression on Lamin-associated heterochromatin. And I noticed that you show that similar effects are also shown in replicative senescent cells. But I didn't see any experiments related to oncogene-induced senescence. I'm just curious. Do you see similar Lamin related effects in oncogene-induced senescence?
Yeah, we do. These are also, again, seemingly the story emerges that these are kind of widespread features, is that this sort of downregulation of this particular Lamin B1. And LBR is essentially, LBR stands for Lamin B receptor. So it's basically the anchor in the nuclear envelope membrane to which Lamin B1 connects, right? And so in addition to that, to which also the chromatin, heterochromatin, connects. So there are kind of multiple sort of docking sites, but that's basically the anchor, one of the anchors that sort of scaffolds overall the genome at the nuclear envelope. And so imagine now LBR going down, Lamin B1 going down. They actually do go down in oncogene-induced senescence as well as in replicative. So that basically compromises, right, one of the anchors for 3D organization. So again, with consequences that we already sort of talked about. And the interesting part is indeed, so it's not, again, mechanisms how these two proteins go down are being investigated. There are, once again, multiple. Apparently, it happens at the RNA level, so the RNA coding for both actually goes down. It also happens at protein level. Well, if the RNA goes down, of course, the protein level will also go down, but that's not the whole story. It can also regulate protein stability, and that seemed to be happening as well. So interestingly, WRN somehow achieves the same downregulation of basically level of Lamin B1 and LBR are about half as high in Werner deficient cells as in normal cells. The only difference being absence or presence of WRN in this particular model system. So what we found was that, again, half of that or some of that effect is transcriptional. And seemingly WRN is involved in supporting kind of normal level of LBR in the cells. And that is a transcriptional function. So that's what we call canonical function, because it sort of is expected. And we showed that it's probably, well, we have evidence suggesting that it probably has to do with WRN canonical function, actually, of mitigating or kind of eliminating secondary alternative structures on DNA, such as R loops we mentioned in the paper, and or G quadruplexes. So again, this is something that that is sort of predicted by prior data and does involve WRN enzymatic activities. But that's one thing. And so we're talking about kind of the canonical function of WRN in controlling transcription, actually, of LBR gene. So through the potentially, again, from our what our data suggests, it's potentially through kind of eliminating, unwinding, or degrading R loops that are actually able to form on the LBR gene. And so, or DNA G quadruplexes. And of course, here I have to define, right? So what is R loop, right, for the audience?
And what is quadruplex as well?
Yeah, so what the R loop is essentially, imagine it's an area where DNA is parted, so melted, and one of the strands is actually hybridized with RNA, and usually it's the RNA that's been transcribed in this region. So it has the hybridized kind of RNA DNA hybrid part, and also has a tail that sort of hangs. And typically that shouldn't be happening. Typically RNA should be just processed away. But in case of R loops, kind of these abnormally stable R loops, that part sort of remains. And that can actually pose a problem for transcription itself, as well as for replication. If now replication encounters this kind of R loop, it also has a sort of a problem proceeding. And so WRN actually was known to enzymatically unwind or degrade R loop. G quadruplex is essentially another alternative non-double helix DNA structure that certain sequences can adopt. And it's essentially basically it's a quadruplex. So it's a 4 strands. It's kind of a same strand DNA that can actually fold, in this case, 4 times around each other. So forming kind of this quadruplex rather than duplex. And so one feature of G quads is they oftentimes form, they actually come together with R loops. For example, that melted part of and in fact in LBR genes, the regions that form those overlap largely. So imagine, right, I told you about the melted DNA duplex is melted, so one strand is hybridized with RNA, the other is free, single-stranded. So it can actually adopt, if it will adopt G quadruplex configuration. And so that creates kind of a double obstacle now. And WRN is actually suited to deal with both. So this is remarkable. So that's a perfect case where WRN can actually regulate gene transcription by helping kind of mitigate these structures. So,
sorry, and in which cases do they normally form with R loops and G quadruplexes?
Yes, so a very good question. And the answer is, in brief, there probably are many conditions and that they would form that are kind of not really not all well understood, not fully understood, because what is the easy one I could say, well, conditions that favor G quadruplex are, for example, where this a DNA sequence that can actually form this has to be G rich with a certain periodicity, sort of going in rich, right? So when this sequence is in single-stranded form and somehow is kind of pushed into single-stranded form, these are the preferred conditions. Then it can actually G quadruplex can form. So that's kind of one case, sort of very easy to understand. So in terms of R loops and RNA DNA hybrids, I guess again, kind of trying simplistically, the understanding is that they are sort of a fixture of life. They actually happen. But, anytime transcription happens, you can sort of have an R loop. But generally speaking, they're short-lived, because they're actually, in addition to WRN, there are many other enzymes that would actually make sure that R loop is not sort of formed. So it's really about kind of regulatory milieu in the cell that actually can result in, or a balance, dynamic balance, that can result in elevated or reduced level of R loops throughout. So I guess I would say that now there's kind of more appreciation of the fact that R loops itself can actually perform regulatory functions. So it's sort of there might be conditions where you might want to keep it, and it will achieve, for example, at the ends of genes, it might play actually a kind of regulatory role, excuse me, in termination, for example. So it's basically, it's not all bad. It's just when there's sort of too many of them, they stay for too long, they may be too, or maybe too extensive, is when kind of things begin to be disruptive. So that's my long answer.
Cool. And basically, these canonical functions of Werner helicase help to deal with the structures, and this prevents genomic instability in a way. But also, it also helps to potentially regulate some genes like LBR, right?
Absolutely, because again, if there's too many of them, stay for too long time, what happens is, rather than kind of unwinding them properly and keeping DNA intact, you might actually address them by breaking DNA and kind of excising that whole R loop or that whole G quadruplex. And that becomes a DNA break, and that becomes DNA damage, and that becomes a potential source of genomic instability. So that's how elevated R loops result in sort of genomic instability, for example, or G quads result in genomic instability. So.
And in your paper, you also show that non-canonical functions of Werner helicase can actually potentially regulate the LBR function. Expression, right?
Absolutely. So that was sort of really kind of, I think, very interesting finding. And specifically, it is that it is about LBR. And the thing about LBR is it is essentially, it's, well, of course, I mentioned it's a nuclear envelope anchor. So what it means really, it's a transmembrane protein that that has a kind of a membrane anchor in the inner nuclear membrane. So it faces into the nucleus, into the nucleoplasm, but it's anchored in the membrane. So as such, it is essentially, so let's just put it this way. How do transmembrane proteins actually get into the nucleus in the first place? Well, they get there from the outside of the cell, where they are synthesized in the endoplasmic reticulum. And so then from there, they sort of travel through the nuclear pore and into the nucleus. So oftentimes as a result, and that's because ER, endoplasmic reticulum, is actually contiguous with the nuclear membrane. So oftentimes as a result, such proteins have some presence in the ER, right? So there will be sort of a cytoplasmic portion that is actually anchored in the ER, and some other fraction of the protein, population of the protein, will be in the nucleus. So LBR is one of those that is typical. Well, oftentimes you look and it's mostly in the nucleus, but there will be situations where you see, some of it is outside the nucleus. So it's sort of on the ER. So apparently, if you don't have WRN in the cell, you actually exhibit more of that cytoplasmic LBR. So there's essentially redistribution of the localization of LBR from predominantly nuclear to kind of mixed. And so that is really remarkable because essentially it obviously reduces, once again, effective concentration of LBR in the nucleus where it can, it performs its function of anchorage of the chromatin. And so, but the question is how, and that's really kind of the reason that we called it non-canonical function of WRN is because it's non-enzymatic. So basically enzymatic features, functions of WRN are not involved in this effect on redistribution. So it has to be some kind of a protein-protein interaction between WRN and some other protein that actually achieves that kind of regulation that results in this distribution. And so that's really interesting in a way, and could there's sort of a lot to look into there in terms of the mechanism.
Thank you. Very interesting. And as I understand, you also speculate that that since Werner helicase can regulate expression of LBR, and it could be different in different cell types. This could lead to manifestations of Werner syndrome in particular tissues. So can you elaborate more on that, please?
Yeah, so that was that sort of came from, I guess, the LBR and Lamin B1 observations. And behind it is the fact that unlike sort of Lamin A, Lamin B1 as well as LBR expression in levels are actually variable between sort of tissues of different origin. And there are some tissues, and that's again work by many labs sort of that's preceding the kind of the set of observations that that went into the paper. And so there are cell types, there are tissue types where different anchors, meaning LBR versus other anchors, of which there are sort of several, different chromatin anchors and different lamins are utilized or are dependent on differently. In other words, there will be cells which with very low level of LBR and low level of Lamin B1 and or low level of B1. So these cells are fine. They just don't utilize these proteins for their sort of chromatin anchorage, for their nuclear kind of support. And so then the speculation came that, well, potentially, what if in these cells, because of how they kind of run their 3D genome, if you'd eliminated WRN from these cells, it wouldn't really matter that much because LBR level is already low and cells do not depend on LBR, for example. And but there will be other cell types that really do depend on LBR and Lamin B1. And therefore, eliminating WRN from these cells would actually critically reduce the LBR and Lamin B1 levels in these cells. And so these cells will have kind of be affected more. So that's really the hypothesis. And the reason actually to hypothesize this and to think about it is that it's been known from the very beginning that Werner syndrome is remarkable in a sense that how sort of different tissues are affected differentially. For example, generally the sort of the ectoderm, skin, neurons are not affected as much in Werner syndrome as some other tissues, such as adipose tissue, for example, or actually the sort of the actual the kind of the dermis and the other tissues that are formed by fibroblasts. So that's people say that's just overall to summarize, it's what they say is a mesenchymal lineage is affected. The mesenchymal lineage that makes makes fibroblasts, makes muscle, makes adipose, makes cartilage, and so forth, is affected more than the than ectoderm and endoderm that basically make that make skin, for example, and gut. And neurons, right, the nervous system. So these are the types of tissues that are affected less. And so I'm currently thinking, and again, it needs to be experimental verified, and more work is needed. But the hypothesis is that maybe the source for tissue-specific manifestation of Werner syndrome has to do, at least in part, with the way, with this kind of linkage between WRN and LBR and Lamin B1. So.
Great, thank you. Also, as understood, there are kind of two stories. One story is about this interaction between HP1α, HDAC2, and Werner helicase, which is downregulated in senescent cells, but also my question, how is it affected by Werner depletion?
Yeah, so in a way, kind of this is where the story started. Basically, for me, the story started in Werner and HDAC2, which is histone deacetylase 2 interaction, which we sort of discovered in earlier work, and so and that's kind of where we kind of got from that interaction that we discovered is actually decreased in senescent cells to kind of probings of different complexes between HDAC2 now and HP1, WRN and HP1, HP1 alpha specifically, which is heterochromatin protein 1 alpha, which is a very prominent constituent of constitutive heterochromatin. So in all of these cases where we're sort of probing all these pairwise, if you will, protein interactions, we were finding decreases of these interactions that are actually structural components of, or structural sort of underpinning of heterochromatin in Werner deficient cells or in senescent cells. And moreover, we found that Werner is physically, of course, part of these complexes. That's where comes kind of this, the structural, the protein-protein interaction role of WRN in heterochromatin. So it's physically present there. It's participating in these interactions. And without it, these interactions are all reduced. So, so that's really kind of where this kind of led from there to probing the interactions, for example, between HP1 alpha and say LBR or Lamin B1, kind of connecting from seeking connections from these structural elements of heterochromatin to kind of these anchors in the membrane, and that's how we progress to looking at LBR and so forth. Now, I should say that again, we were not like absolutely first, certainly probing these interactions. We're not absolutely first. An earlier paper, it was actually in 2015, very prominent paper by kind of this international collaboration of scientists, noted features of heterochromatin loss in Werner deficient, actually mesenchymal stem cells. So many of these deficiencies were described in that paper, and were actually pioneering introducing this concept of loss of chromatin, of heterochromatin as kind of aging, as something associated with or causative of aging in the cell. But what we've contributed in this particular work is essentially, the question for me always was, well, in mesenchymal stem cell model, these cells are senescing. They will be senescing whether they have WRN in them or not, actually. WRN positive mesenchymal stem cells will senesce. It just will take them a little longer. And so, and if you don't have WRN in mesenchymal stem cells, they will senesce faster. So what I always wanted to do was to uncouple this kind of very strong preponderance or sort of preponderance towards senescence in this system, uncouple it from the study of WRN effects on heterochromatin. I wanted to see, well, in the cells that do not senesce, the cells that proliferate, can we still see these isolated, in this isolation, these WRN effects? And that's what this paper does. It says, well, even if these are proliferating cells, you still have these direct effects of WRN on heterochromatin. And that's really, and then we sort of trying to kind of see whether we can trace these effects to known functions of WRN, such as in replication. And the answer is that no, not all of the effects on heterochromatin can be explained by old, previously known functions of WRN. There has to be something that works directly with heterochromatin. That's kind of the other, basically, new message or new take-home that comes from this work.
Thank you very much for clarifying. The next question I would like to ask is, do we know how Werner helicase transcription changes during aging?
Oh, yeah. You mean Werner itself, the gene transcription?
Yeah, expression. Like, do we see decrease in Werner?
Yeah, it's actually, so at the transcript level, I actually don't even know, I must say, I must admit, confess. But what I do know is if you look at the protein level, you sometimes see reduction of the protein level in aging cells and in senescent cells. Not always, but sometimes. So there is, which of course will sort of suggest that maybe cells use WRN as natural means to control their own senescence program. So basically, it sort of feeds onto WRN. But that's again, that's a kind of a speculation.
And do you think overexpression of Werner helicase could improve or extend lifespan potentially?
You mean improve
overexpression?
Oh, right. Oh, the overexpression. Yeah. So that is a good question. And I think my short answer is I don't know, but I don't think so. And that is because essentially because the functions there are sort of the regulatory kind of inputs are somewhat buffered and complex. I don't think that just making more WRN will just sort of improve things, will shift these processes, will result in more LBR or more Lamin B1 or more of all these protein-protein interactions. It's just kind of, I don't think that would really have a huge shift, sort of dynamic shift on the whole system. What I do know is also that just from a technical point of view, WRN overexpression is sort of hard to achieve in terms of kind of engineering. It basically, it will, again, buffered systems, so there will be more degradation. So essentially you try to force it up and it's actually going to be degraded more. So there's kind of that technical, if you will, sort of engineering problem there because of all these, because it's really, everything is tightly controlled. So it's easier to elicit the effect by just taking WRN out of the system than to kind of pushing more of it on the system.
Mm-hmm.
So that's the answer, I guess.
Thank you.
Mm-hmm.
And I'd like to ask just general question. What do you think the main mechanism of aging, in your opinion?
Oh gosh. Well,
you might be biased, I guess. But...
yeah, so that's sort of putting me on the spot there. And well, I wish I knew, right? I wish I had kind of a very simple answer. I never really, I think there are no simple answers to this. It seems, of course, my personal preference is this kind of heterochromatin loss and resulting sort of these effects, but that's not the case. I guess if you look at the hallmarks of aging as defined in the field, there are, let me try to sort of recap them so basically heterochromatin loss, genomic instability, reactive oxygen species elevation due to mitochondrial dysfunction, loss of proteostasis, that means autophagy primarily. So basically you're not able to kind of recycle your protein components as a cell. And there are others such as cellular communications are sort of disrupted. So as far as I remember, these are the ones. And you have to admit at this point that all of them are important for in the process of aging, all of them. And sort of so there's no one thing that kind of does the normal aging. There are situations where one of these hallmarks is kind of exacerbated by a particular condition. For example, massive DNA damage will kind of outsizedly have an influence on aging. But it doesn't mean that normally any of them is not important. For example, you can have, it is now appreciated that, for example, patients surviving chemotherapy, essentially, there are signs that kind of the aging is accelerated, but that's because of that massive level of DNA damage. That's where DNA damage predominantly kind of affects it, but it doesn't mean that it's an exclusive factor normally. So really the important other question is whether all these hallmarks are kind of independent, sort of just silos of processes that just impinge on lifespan, or are they all connected? And I think the answer now is that most of them are really connected in these intricate, so one kind of leads to another, or impinges on another, or facilitates another. So they're all just sort of interconnected this way. And then, of course, other question becomes, if so, that maybe all of them are just manifestations of something, one single thing that is at the base of all of them. And we don't know what it is, and we don't know whether it is there, but that's one way of thinking. And another way would be just that, yeah, there are all these hallmarks, all these processes, and they all kind of work together to bring us aging.
Thank you very much for your interesting paper and for sharing your insights. And I hope to see your new studies soon. Yeah.
Thank you. Yeah, that was a great pleasure.
Thank you. And
thank you.
Cheers.
Have a great day. Have a great night.